This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 913, recorded on June 24th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent and the gang. Looking at my window, it's puffy white clouds, gentle breeze, 82 to 84 degrees, humidity around 45 to 50%. In other words, a perfect summer day.
Perfect. And not a lot of humidity.
No.
It's very tolerable. Also joining us from Western Massachusetts, Alan Dove.
It's great to be back on a Friday TWiV. And here in Western Mass, it's 79 Fahrenheit, 26 C, and another glorious, beautiful summer day and what has been a gorgeous early, an atypically gorgeous early summer here in Western Mass. It's just been lovely weather, and we haven't had the heat or the AC on. We just open the windows, and it's wonderful.
From Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 85 Fahrenheit, 30 Celsius. And it says that tomorrow we're going to have an air quality alert, but I'm not sure why. Anyway, it's
the temperature's going to go up
80, just to 87. So it's a little warmer than my favorite, which is 82, but still pretty nice.
Right.
And from Austin, Texas, Rich Condit.
Hi, everybody.
Tell us your troubles, Rich.
It is 99 degrees here now. We're headed for, I think the forecast is for 101 today. Yes, forecast for 101. We are working on day 19 of temperatures in excess of 100 degrees this year.
Wow.
We're moving rapidly towards a record for June number of days over 100 degrees. So it's hotter than stink here. But, it's okay. Yeah, I went out for a walk this morning. It was quite pleasant at 6:30 in the morning.
Yeah, it's a good hour.
This is the part of the year where we get to have the better weather than you, Rich.
Exactly. Exactly.
So speaking of the gang, as Alan said, I had occasion to fire up Skype this morning.
Oh, wow.
Which I hadn't done
Did
in a
it
long
sort of
time.
go clunk, clunk as you fired it up?
I actually logged in and everything. But I've done a couple of Skypes with some people that just use Skype. But I saw some of our old Skypes from, they still have a list of all the recent, quote unquote. It goes all the way back to 2019 with Kathy, Alan, Rich. We were all on Skype at the time, remember?
Yep.
Yeah. We used to do just audio.
Skype for years.
Yeah.
We didn't see each other on Skype, but now there's of course a video component. I didn't have a webcam for all those years, right?
Actually, wow, that's really, I can hardly remember not seeing each other.
Yeah.
Is that right?
Yes.
Cow. I guess
Except
that's
for
right.
Alan.
So the way I used to do it was I had one Skype computer for everyone. Everyone would log, and I had a different Skype account on each computer. Everyone would log into a different Skype account. Then I would mix them all in the studio and record them. And so we didn't need a video. But then we got sent home, and I didn't have 4 computers at home. So we had to use Zoom, which has many limitations. But one of the ups is that you can do videos. So I got webcams, and here we are.
Yeah.
Hard to imagine doing it without video now.
Yeah. Yeah. I was just thinking the audio podcasts seemed like they were just a completely different activity.
Yeah.
They still get a lot of downloads, the audio only.
Yeah.
In fact, more than the video.
Really?
Yeah, consistently more than the video.
I wouldn't want to sit there and watch us for 2 hours.
No.
On a podcast, you can walk around.
Once is
that's
enough.
the magic
Rich,
of radio is you can multitask.
Yeah, many people walk around and do things.
Magic of radio. That's
Many
right.
people do watch, and they like
Yeah.
to see who
By
we
the way,
are.
for those who are watching this episode, have you done like a little public service announcement about that plaque in your background, Vincent?
We
Oh,
did
yeah.
last time.
Okay,
Yeah, we
yeah.
explained it last
Yeah.
time. Yeah.
Don't try to click that button.
Yeah.
That's actually an object in Vincent's office.
Yeah, that's a very nice. We have 110,000 YouTube subscribers, which is really cool.
Great.
I'm very proud of that. And that's all, pre 2019, we had 20,000 subscribers on YouTube. And I don't know if you remember, YouTube had a contest or a
Yes.
promotion where they said we can give you funding for your program if you can get over 25,000 subscribers. And we appealed, and we could not get.
Couldn't quite hit it.
Ah,
But a virus did it.
Yes.
it sure did.
And I have to say that to this day, when I see what SARS-CoV-2 has done to the world, I'm actually still stunned
It's
at the
amazing.
havoc it wreaked, right?
Yeah.
Still
It
reeking.
is, and we didn't really, in our lifetimes, we didn't see this. I suppose 1918, it was similar.
Yeah.
Still wreaking havoc, permanent changes in everything. So many people died, but then so many people's lives altered. It is really, really amazing. Okay, so we have a few things to talk about today, including an update on which I thought it's good that Alan is here because he will enjoy this.
Yes.
This is the finding of poliovirus in the sewers of London sewage. And this has been all over the news. And, it adds to this frenzy of viruses. We have SARS-CoV-2, we have monkeypox, and then poliovirus makes the news. Hey, I thought that was taken care of. But, many countries do routinely monitor sewage for the presence of poliovirus. And in countries that have switched to the inactivated vaccine, that would, of course, really reduce the amount of oral poliovirus vaccine, which is infectious, that you would find in sewage. And in this case, what they say is, we do detect OPV in sewage from time to time because people come from other countries where they still use OPV. Quite a few countries still use OPV, and they shed virus, and it gets into the sewer, but then it goes away. But this has been consistently seen now for a few months, and apparently the genome sequence is changing, suggesting that it's
Suggesting community
reproducing.
transmission. Yeah.
Yeah. And apparently vaccination rates have fallen below 90% in some London communities, which is not good because that you can get infected and potentially get paralysis. So when you use oral polio vaccine, you ingest it, it reproduces in your intestines, and it's shed, sometimes for months. But it also undergoes genetic change in the intestine and it acquires the property that it lost during development of the vaccine. It acquires neurovirulence. And so these are called vaccine-derived polioviruses, and they can circulate for years. They can cause outbreaks of poliovirus in communities where vaccination rates drop. So that's really the concern here.
And I
assume that when you're
I believe it's the only vaccine in regular use that can cause the disease that it's supposed to prevent. Is that right? Is there any other that?
That's what I've read. Yeah.
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 So, am I correct in assuming that when you're like monitoring sewage and that kind of stuff, you can distinguish between the vaccine-derived polio and other stuff just by sequence? You can see some of the attenuating mutations, but not all of them, or something like that.
Yeah, you can distinguish vaccine-derived from wild-type poliovirus. Yeah.
Well, the virus survives very well in sewage, which is its primary mode of transmission.
It's infectious. It's not like SARS-CoV-2 where they're just bits of RNA.
Yeah.
This is infectious virus, yeah. And now the other thing is that when you use IPV, your intestines are still susceptible to infection.
That's inactivated poliovirus.
Inactivated
the
vaccine.
Regular virus has been chemically inactivated.
Yeah, so these OPV, these vaccine-derived strains can circulate for quite a long time, quite silently, even in a well-immunized population if they've used IPV.
Yeah, so the IPV prevents you from getting the paralysis very reliably, but it doesn't prevent you from carrying the virus through your intestines. Of course, if you then subsequently get vaccine-derived polio and it replicates in your intestines, now you have immunity in your gut because you've gotten the secondary immunization with the virus. But then you provided more to the sewage.
So, the solution, there are two potential solutions to this. One is, this is a global solution. One is to switch everybody to IPV, inactivated vaccine. But it's hard because it has to be injected. And, the OPV you just drink, it's so easy to give, right? And you don't need any trained personnel to give it.
Is there more of a cold chain issue or not for the inactivated?
I think it's the same. I think it's the same issue.
Okay.
The other is to make OPV that don't revert. And we actually talked about that work, last year or 2 years ago. There is a new type 2 OPV strain that's been developed, which seems to be less likely to revert, but that has to be put into people before we find that out. We're still immunizing because there still is wild-type polio in Afghanistan and Pakistan, so you still have to immunize because as long as there's wild poliovirus around, and then the problem is that if most countries are still using OPV, you still have circulating OPV-derived strains, and if you let immunization drop, you have outbreaks. In fact, if you go to polioeradication.org and you look at the current situation, you will see lots of orange dots all throughout Central Africa, which are outbreaks of type 2 vaccine-derived polio in communities where immunization has dropped.
Yeah. And this is, so Afghanistan and Pakistan have been having this ongoing problem, and it occurs pretty regularly. And in fact, I think, wasn't Nigeria having problems again? It's been very hard to eradicate in Nigeria. I know that.
They had a number of problems, and there have also been some other. So that's type 2 OPV,
right.
And there have been some other serotype outbreaks as well. And even wild type viruses can cause outbreaks because people travel from Afghanistan and Pakistan. They can bring the virus with them, and if you're not immunized, you can get paralysis. In March there was a case of paralysis in Israel. 3-year-old child who was unvaccinated and got a vaccine, type 3 vaccine-derived virus. So,
this is something where the pandemic, the SARS-CoV-2 pandemic, is making this more of a risk because a lot of the vaccination campaigns, especially in poor countries, got suspended during the pandemic because they couldn't get the people where they needed to go, and you had to have everybody locked down. And so there's been a drop in vaccination coverage across the board for all sorts of childhood vaccines, especially. And there's concern that this sort of thing is going to bust out again.
But as long as you keep vaccinating, in the case of the UK, make sure your kid is vaccinated.
Yes.
And you're not going to have an issue. So we don't actually look for polio in the sewers in the U.S. And why not? Well,
because we might find it.
We might find it, and then
that'd
you have
be horrible news.
And Mark Palanch, who used to be at CDC, I once asked him, "Why don't we look?" And he said, "Because then we're going to find it, and then we're not sure what to tell people." And you can see by this situation, they told people, right? They have a good surveillance system, which is good. It picked it up, but we don't. And as long as most people are immunized, it's not a problem. There are two articles I'll link to. One is a ProMed Mail article, and then Alan, I think, must have put in the Science article.
Science.
The Science article is good. There's one point I want to just say is not quite correct. At the end of the article, they're talking about OPV, and it's written, although it occasionally evolves to regain its virulence. It's not correct. In every person who gets OPV, the virus reverts in the gut, and that you can isolate neurovirulent virus from every child after just a few days.
Yeah.
So it's not occasionally. Occasionally it causes paralysis.
Yeah.
Like 1 in 1.5 million doses of vaccine, but it's always reverting.
Right. And occasionally somebody gets the revertant in their feces and it spreads to somebody else, and that causes paralysis. But
this goes to show how poor our hand hygiene is, right? This is all fecally contaminated material.
Yeah.
Dixon, you were going to say something?
Yeah. What about the sanitary engineers, so to speak, that are taking care of all those water treatment plants? I wonder what the surveys would show on them.
Well, they're all
My guess
vaccinated,
is
right?
that they're pretty careful about. I mean, if you work around sewage all day, you'd kind of live in protective. I would kind of live in protective equipment throughout the day if that were.
I'm sure
Or
they
maybe
do,
you're
but
maybe you're constantly getting low levels
Or
of
you're constantly getting
immunization from
immunized, yes.
exposure. Yeah.
there's a lot of aerosolization
Sure.
technology which could put the virus in the air instantly, obviously.
I have very high titers of antibodies to poliovirus.
Yeah.
Yeah, from having worked with it for years.
I want to hear what Kathy has to say about
Yes.
the
Well, so it was
Jewish
in this
word.
ProMed Mail, which was not the first thing that I read about it, that points out that it was the sewage treatment plant was the Beckton Sewage Treatment Works in the London borough of Newham, East London. But this is the fun part. It was formerly known as Barking Sewage Works. And for some reason, they've changed it from Barking Sewage Works to the Beckton Sewage Treatment Works.
I like barking.
Yeah.
Yeah.
I'm looking at it.
Barking is probably like the name of the location historically,
Yes.
and then they
Yeah.
decided to name it after somebody, which is a dubious honor.
Yeah, it's right on a creek. I'm looking at it on Google Maps. It's right on. It's a huge facility. It's right on a creek called Barking Creek. And there's even a Starbucks right across the street.
Of course there is.
That is a creek you do not want to go up without a paddle.
Yeah.
So, as I said, we get a lot of emails about this, but one from Justin, he linked to an article and he wrote, "You and Alan said this years ago." Remember, Alan?
Yes.
Yeah.
We
You
said.
should-
Go
Yeah.
ahead.
Yeah, I was going to say you should put the link to that science commentary in also. That
Yes.
PubMed science commentary. I mean,
So that we wrote a commentary in 1997, Alan and I, and it was published in Science. The polio eradication effort. Should vaccine eradication be next? And, we said, you're going to have troubles if you stop vaccinating because there are all these circulating vaccine-derived polioviruses, and you don't know how long they can circulate. And I think there was a rebuttal from WHO.
There was a rebuttal from, I think it was somebody at CDC or WHO, which basically said, nah, that won't be a problem at all.
No problem.
It's
There's
just
time
kind of
limited.
hand-waving away the problem.
They said there's time-limited circulation of these. And then within the year, we learned that they can circulate for decades.
And
Not
then
that we knew.
within a few years, we had vaccine-derived outbreaks popping up in places where they thought they'd eradicated it.
But, to be fair, you and I didn't know, but we were just being cautious, right?
Yeah. And I don't really gloat about this because
No,
I don't
not at
feel
all.
good about having been right. It's one of those where, I hope we're wrong about this, but this is a concern. And then they was like, oh, no, not a concern. All right, well.
So maybe you already said this, and I was goofing off with Google Maps, so I didn't hear it. But I mean, the implication of the sort of dynamics of this is that somebody is shedding this over a period of time, or possibly, okay? Because usually it shows up and goes away, but this has been lingering. So it'll be
Probably a
interesting.
few people. So you figure, so one person comes to the UK, right? They've got vaccine virus in them. They go to the toilet, it gets into the sewage, they detect it, and then it's gone in a couple days. But the suggestion is that it's somehow spreading to other people because it's persisting a long time. I don't know. I guess it could be one person who's infected for a long period of time, right?
It could
I
be.
suppose. I don't know.
It could be a combination of these things. One person infected for a while and also secondarily infecting others, especially since the people who are being vaccinated are generally children who have horrible hand hygiene and are extremely good at spreading fecal to oral pathogens. So this could be just spreading among a group of kids in a school somewhere, and it's showing up at the sewage treatment plant.
Well, and that's the I wouldn't say scary, but the notable thing is, and I think that's what we want to know is this actually spreading in the population?
Yes, right.
Oh, I would not be surprised if it were, because with an IPV immunized population, the intestines are fully susceptible.
Yeah.
You just don't get disease, right? So you get infected, you shed virus. And so someone has asked me, then even if the world switched to IPV, we'd never get rid of these vaccine-derived strains. It's possible we would not, because they'd always have intestines in which to reproduce.
But I suspect they would burn themselves out if we're not constantly reintroducing them.
Hopefully.
Eventually.
Yeah, if we could stop using OPV.
Right.
Now, the interesting aspect of this is that a number of years ago, we stopped using trivalent. We removed the type 2 component because that was the first to be declared eradicated. So now they're immunizing with divalent types 1 and 3 only. So where do these type 2 outbreaks come from? Whenever there's an outbreak, they go in with trivalent to mop up the type 2 cases because it's really good at limiting an outbreak. And that reintroduces type 2 OPV, and then you're back at square one.
Yeah. But if we do get to a point where we're vaccinating the whole world or most of the world with IPV, if we look back at the history of polio before the vaccines, it did tend to be seasonal, at least in the northern and far southern hemispheres. So there may be a crash of the vaccine-derived polio in the population to the point that you could actually see it disappear.
Yeah, I think when they go in now and mop up, they are using the new OPV2. I think. I'm not sure because it hasn't been officially announced. I've just heard people saying it's already been in millions of people, and that could only happen if they're using it for mop-up, because the trials didn't involve that many people. And so maybe that will limit it. We'll see.
Yeah.
Because that has very specific changes in the genome that you could detect. You could say, this is NOPV, and guess what? It's circulating or it's not circulating.
Right.
So we will see. All right. Now, I wanted to talk again about long COVID. We did a paper a couple of weeks ago. I think that was your last time on, Alan.
I think it was, yeah.
It was a VA study where people going to VA hospitals were looked at. And this one is a different study. And I wanted to talk about this very short Lancet paper. And then there's an accompanying commentary in Nature that I wanted to bring up as well. So the Lancet paper is risk of long COVID associated with Delta versus Omicron variants of SARS-CoV-2. So the question many people ask is, okay, we have long COVID. We're not quite sure of the frequency because, as you'll see, every study has a slightly different number. But is it any different when you compare Omicron versus Delta? And they say in this...
I was going to say, speaking of London, the authors are Antonelli et al., ending with Claire Steeves, and they all seem to be in London.
Right. Yes, that's right. And they do say here that the in Europe, the number of Omicron cases exceeded all previously reported cases. A lot of numbers there. So if Omicron has a high long COVID rate, a lot of people are going to end up with issues. So this is a case control observational study. And what they did was they have self-reported data from the COVID symptom study app, which was developed, I guess, at King's College. And so it's all self-reporting. You go to your app and you tell them how you feel. I guess they have a number of different symptoms that are consistent with long COVID, and you can tick them off, tick the little boxes, I suppose. And these are symptoms 4 weeks or more after the start of acute COVID. Now, that already is an issue because WHO just recently redefined this to be 8 weeks, because I think a lot of it, I asked Daniel the other day, he said a lot of it resolves within those 4 weeks.
Yeah, that's a big difference between 4 and 8 weeks.
So I suspect the numbers that we see here are going to be even lower if you redid this for 8 weeks. And then they were able to have cases in two distinct periods which correspond to Omicron versus Delta. So the inclusion criteria, positive SARS-CoV-2 PCR or lateral flow antigen test after being vaccinated, at least one log per week in the app for at least 28 days after testing positive, and no previous infections before vaccination.
And by one log, they mean one entry in the app.
One entry, yeah.
When I first said one log, I was thinking a factor of 10.
Yes.
What?
Yeah. Yeah, entry would be a better word, right? Yeah. All right, there's 56,000 UK adults testing positive between December and March, December 21, March 22. These are referred to as Omicron because most of the cases at the time, over 70% in the UK, were Omicron. But they didn't test. They didn't look at that, so that's a weakness. Then they identified 41,300 UK adult cases between June 2021 and November 2021, which they referred to as Delta because, again, most of the cases were Delta. They have both symptomatic and asymptomatic infections. All right, so now the goal is to look at the association between long COVID, that's the outcome, and the infection period, which is exposure. And they use statistical procedures to do that, and they group the data according to time elapsed between infection and the most recent vaccination. So we have 3 months, 3 to 6 months, and more to 6 months. And they say we did this to allow for any effect of the reduction of immunity from vaccination.
So this is all in vaccinated individuals.
This is all in vaccinated individuals, correct.
That was part of their inclusion criteria. Why? I don't understand why. Unless the app is kind of like the VSafe app where you get it when you got vaccinated to report the sort of what's going on after your vaccination. I don't quite understand that. I mean, it does make for a little more consistency in the group, but there was no clear justification given for why you would choose a vaccinated population, at least that I saw.
So my understanding is that this is the main concern because people say, well, why should I get vaccinated if I'm going to get long COVID anyway, right? I mean, there have been studies of long COVID in unvaccinated populations is much higher. But if you want to assure vaccinated people, right, this is the study to do.
Okay.
Yeah, this struck me as the real headline question. If I'm fully vaccinated, am I still going to get long COVID? And that's really what they're trying to address here. If you're not vaccinated and you're getting long COVID, that's a separate ball of wax.
And of course, Delta versus Omicron is a big one, right?
That's the big question.
Oh.
Yeah.
So among Omicron cases, 2,500 out of 56,000 people, that's 4.5%, experienced long COVID. And, that's a very short sentence, but it's really important because I would like to know exactly what symptoms they all had, right? And you never get those data. And in fact, the VA study, how many people just had fatigue? How many people had?
So you can expand the sentence to say among Omicron cases, 4.5% of people experienced self-reported long COVID.
Yeah, that could be. All right, so 4.5% for Omicron, and among Delta, 10.8%, 4,400 out of 41,000. All right,
twofold difference.
Twofold difference, and that's the overall among all the cases. And then there's a very nice figure where they graph.
There's some debate about the niceness of this figure.
So basically, those numbers. So remember, they stratify it by vaccine timing: greater than 6 months, 3 to 6 months, less than 3 months. And they say Omicron cases were less likely to experience long COVID for all vaccine timing. And they also calculated odds ratios for long COVID, which ranged from 0.24 to 0.5.
And that must be the chances of getting long COVID after Omicron compared to
the Delta.
Yes.
Okay.
Yeah. Right. So this figure, I Kathy and I, and I think a couple of other hosts, as we were talking before.
I was kvetching as well.
So once you parse this thing, it's one of these figures that they've packed, I think, 5 dimensions of data into this little panel. And it's just some, little lines on a graph. It doesn't look like a whole lot until you start saying, wait, what does that line represent? And you get this conclusion that Omicron seems to have a lower risk of long COVID based on this 4-week window in all of the age groups, although there are differences in the age groups, and in all the vaccine groups, although there are differences in the vaccine groups.
What's interesting is that greater than 6 months, there's no difference compared to less than 3 months. So there's no, in fact, less than 3 months, there's a higher odds ratio compared to, which may be some immunological maturation issue of some kind. I don't know. And that's consistent across all the age groups. They have all age groups. They have 18 to 59 years, and then they have over 60 years.
Right.
So interestingly, if you're vaccinated over 6 months ago, the odds ratio, if you're over 60 years, is not much higher than all the other age groups, all the other ages, which is quite interesting.
Yeah.
But from 3 to 6 months, if you were vaccinated 3 to 6 months previously, you have a huge error bar.
There's this enormous spread, yes.
So those data are not very good. And then it increases, the odds ratio increases if you had the vaccine less than 3 months previously. All right, so basically Omicron, if you consider that these are accurate, Omicron is less likely to give rise to long COVID, although it's still 4.5%, which if you extrapolate to all the cases in the world, that's a lot of people, right?
Yeah.
And I would suspect it's still too high. And remember, some other estimates are 50%, right? Some studies say it's 50%. And, and that's why I wanted to bring in this commentary, which is in Nature, as I said. Anyway, they do note the limitations. Well, first of all, they say this is the first peer-reviewed study to report on long COVID, highlighting that health surveillance using smartphone apps can produce rapid insights, which we have consistently shown are accurate and subsequently replicated. And that's not clear if that's correct. Limitations include that the samples are not fully generalizable to the UK population on account of sex and socioeconomic bias. So there are more women in this study than men. And the socioeconomic bias, they don't really explain it, but not everybody has a cell phone, right?
Or a smartphone where you can install this app.
Right.
And this is a kind of a self-selecting thing. I mean, a lot of people are sort of tinfoil hat types who don't want unnecessary apps on their phones. And I raise my hand in that regard as well. You want me to download it? Who are you wanting me to download this app? What's it going to data mine me for? So there's a selection bias built in.
Okay. Now, the commentary is in Nature. How common is long COVID? Why studies give different answers? And I thought this was actually good. I wanted to point out a few things. So first of all, a statistician, Al Ali, who is at VA St. Louis healthcare system, so the VA system has millions and millions of data points. But Ali points out, the studies, all the studies on long COVID that have been done have different numbers. So the one we just did is, 4.5% for Omicron. Others have said 50%. And Ali says the public does not react very well to saying between 15 and 50%. When you ask, what's my risk?
Right.
It's between 15 and 50%. I think that's a good point. And so why is that? Why are the studies different? And they say part of the problem is the definition of long COVID, which has been linked to more than 200 symptoms, which can vary from inconvenient to debilitating, and it can last months or years. There's no agreement on how to define and diagnose it. So that's a problem. And this other point here, how symptoms are recorded in the claims and electronic records. So they say doctors rarely list a code for every symptom a patient is experiencing. And so that can impact if you're looking at electronic databases, that can be a problem. And then they say some studies rely on self-reporting, which in itself is problematic. They can be biased because people who have symptoms are more likely to participate. And studies that rely on smartphone apps might not fully capture data from disadvantaged communities. I think those are really good points to keep in mind with any study.
Yeah.
They're not general. And one more thing, this is Maimuna Majumdar at Harvard says, the number of data points considered is often so large that we mistakenly assume that these data must be representative, but this isn't necessarily the case. So just because they have millions and millions of records doesn't mean, and the VA is a great example, right?
Yeah.
It's not representative. So I think this is a good commentary.
Yeah, and this is everything we've talked about so far is available for free, so people can go check all these out.
So here's a question for you guys. I got an email the other day from a lady said to me, why do you keep calling it a paywall? If you go to a newsstand, you don't say there's a paywall in front. And it is a good point, right? So I said,
yeah, that's the term applied online.
What else could you call it?
Right, because I think it's an extension of the term firewall.
Yeah.
I see.
So in computer security, you have a firewall that blocks incoming connections. If you have a paywall, it blocks incoming connections until you pay. So that's the analogy there.
I mean, if you want to say it requires a subscription to get the article, it's a lot more words, right?
Yes.
Right. Or you can pay $35 for this single article.
Yes.
Yeah, that's right.
That's quite a wall.
It's a pay-per-view. Is that good? People don't like that either, probably.
Yeah,
pay-per-view.
Maybe.
So it's funny. Then someone else wrote, and this is just to show you what people complain about, which is fine. They say, why do you call it a snippet? You spend as much time and sometimes more on this. Okay.
Well, snippets derived from TWiV, right? That's what Elio called them.
Elio had this idea, just 15 minutes, give the overview, which, when Michael Schmidt does a snippet,
It never happens.
it's never 15 minutes. That's the intent. And I think it's a good idea, but actually, the snippet today was the Lancet. I think we spent 15 minutes.
We're not too far off that.
Not too far from that. I don't know. I kind of like snippet. But you can't please everyone. That's the bottom line, right? Okay, so our paper for today. So we will probably spend 45 minutes. I try to end the paper discussions at 4:30 so we can do a half hour, of email and pics. This is published in Cell Chemical Biology.
Is this a first for us to do a paper from this?
I think so. And this is an open
Chemical
access.
biology.
Yes.
I know.
The heck is that?
Not to be confused with biochemistry, although that would be what I would call it.
Yeah, that's what it is, right?
Yeah.
Yeah.
It's chemistry of cells, and that's biochemistry. But they wanted to use a different name.
Except I'm not sure that this is biochemistry, this paper.
Well, okay.
Biochemistry to me is smaller molecules.
Right.
And pathways and stuff like that. And enzymology.
And mechanistic,
Yeah.
Okay.
And at least one Suffidex column.
Maybe that's why they called it. Yes. Maybe that's why they called it chemical biology, because they wanted to publish papers like this.
Yeah.
Last week you were talking about columns, Rich, remember?
Yeah.
And I meant to mention, I forgot. I threw out so many columns in the last few weeks cleaning out my lab. They were Saul Silverstein's. He had dozens, from smallest to 3 inches in diameter and 3 feet. What a waste! But nobody wants them, right?
I think in another life, if I were to do anything similar to what I did this time, I would like to be a biochemist. Problem is I don't have the imagination. I think it really takes quite an imagination. I don't have that. The virus was always my teacher, always my guide. And in biochemistry, you're out there on your own. You got to create hypotheses and test them.
So is Stu Schumann a biochemist?
Oh yeah,
okay,
big time.
Jerry Hurwitz was a biochemist, but you're
Jerry Hurwitz's byline was "Don't think, purify."
Joan Stites wasn't a biochemist, was she?
No, I would call her a molecular biologist.
Okay. And you're not supposed to define things by giving examples, but to me that works.
Yeah, that works.
All right. So this paper is called Identifying Enhancers of Innate Immune Signaling as Broad-Spectrum Antivirals Active Against Emerging Viruses. And we have, the first author is Gislan Marifi, and then we have two authors at the end who contributed equally, Nathalie Arhel and Sebastian Nisol, and the lead contact is Sebastian Nisol. It's from University of Montpellier and the University of Geneva. And I like this because of the technology, frankly.
Yes.
It really is a cool
This
assay.
is really cool.
And they find some interesting things as well. And one of my bars for a paper is how much will you learn by reading this? And I learned a lot. So I think that's really, to take Kathy's sign off, I learned a lot.
Yeah.
So the idea here is, we have many antivirals that are direct acting antivirals, which means they block some step in virus reproduction, like nucleoside analogs and protease inhibitors. But there are far fewer antivirals that interfere with cellular proteins, cellular pathways, host-directed antivirals. There are some. I can think of one, Maraviroc, which inhibits CCR5. It's an HIV-1 antiviral. It blocks the co-receptor and blocks infection. They're harder to identify because if you hit a cell target, you could have toxicity, right? They'd make a statement here, they are less prone to viral evasion, but I don't think that's necessarily true. Grafeldin is an inhibitor of a cell process that inhibits polio, but you can get resistant mutants. You can get resistance to Maraviroc. So I wouldn't say that they're less prone to evasion. Maybe they could be more broad spectrum. Where they're looking for antivirals that target cell pathways and cell proteins. And they say, well, how about innate immunity, right? It's a really early defense, and it's also important for making good antibodies and T cells. So that would be cool. So then
that was not an original idea with them.
Of course.
Giving people interferon is an old idea, and they talk about this in the introduction that this type of thing has been tried before, usually with not great results.
Yeah, interferon has a lot of side effects, right? Because it induces a lot of genes that have effects on many tissues, and they want to be more specific.
or so.
Yes.
Yeah, and they want to be more specific. That makes sense, and that's one of the things you're going to see in this paper.
Right. So the basic idea, to kind of top level view here, they want to turn on the interferon response, the innate immune response, preferentially in cells that are infected.
Right.
That's what you'd like to do.
Because for years, people with hepatitis C were treated with interferon, and it required many, many weeks of treatment, and many of them stopped because they felt terrible. They got flu-like illness, which is caused by giving people interferon, fever, malaise, muscle ache, etc., brain fog. So the innate immune system requires detection of pathogens by pattern recognition receptors, like toll-like receptors, RIG-I-like receptors. And when they detect an RNA or a protein, then there are a series of protein kinases that phosphorylate, in the end, what are called IRFs, interferon regulatory factors, which are transcription factors that go in the nucleus and then orchestrate the production of all the mRNAs that are needed. First interferons, and then eventually the interferons induce the interferon-stimulated genes, which have the antiviral activity. So there are 9 known IRFs, and 4, IRF 1, 3, 5, and 7, are involved in producing type 1 interferon. And previously, others have shown that IRF 1 and 5 are apparently dispensable for type 1 interferon induction by viruses, and so IRF3 and 7 are considered the main inducers of type 1 interferon. And type IRF3 is always on, and IRF7 is induced by virus infection.
So that drills us down to the strategy of let's induce IRF7, which is a simpler problem.
And then they go on to talk about giving people recombinant interferon. It has modest efficacy, poor tolerability, as we have said, because it has systemic effects. They also talk about synthetic agonists that will, instead of giving people interferon, you can give them an agonist, which will turn on interferon synthesis. And one of them is called imiquimod, which for some reason reminds me of Ahab.
Yes.
The other guy on the boat what was his name?
Well, the Pequod
Ishmael.
Ishmael
the boat
and
was the Pequod.
the boat was the Pequod, and the
Who was the first mate? The big guy.
Queequeg.
Queequeg. Yeah.
Queequeg was the harpooner.
All right, I'm thinking of the boat name. Okay,
Yeah.
Imiquim. Anyway, so these have worked.
Omicron. The boat's name was Omicron.
Imiquimod turns on interferon, but it does so in all cells, right? So again, you're having this systemic issue. So can we make something that, as Alan said, is in cells where the virus is reproducing? And so that's what they're looking at here. And what they do is develop a system to measure the movement of these IRFs from the cytoplasm into the nucleus. Because when a pattern recognition receptor, let's say RIG-I, detects some double-stranded RNA, it then initiates a series of events where the IRF goes from the cytoplasm into the nucleus. And then that stimulates the production of interferon mRNA. So they want to measure that step. And it's a very clever system.
Right. So the idea is if you can monitor that, then you could throw drugs at the cells and find one that causes that response. Right. And so they want an assay that is automatable, that they can do easily in high throughput, and then they can look at potentially thousands or even millions of compounds, although in this paper they don't go quite that big.
This paper is like a proof of concept.
It's a proof of concept,
yeah. The system is called Alpha Centauri.
Yes.
Actually, Alpha Cen. And that could make it into the title. You never know.
It could make it into the title, yes.
That's the closest star to Earth.
Yes.
Oh, thank you. How many light years away is it?
4.
4 light years?
Jeez, that's just around the corner.
Yeah, exactly.
Wow, that's pretty close.
You could send a mission there potentially.
It's probable it also has planets.
Yeah, we've seen planets around it, and I don't know. I don't know if we have an Earth-like planet or anything in the habitable zone that we know of.
Probably.
That'd be too
Dixon,
easy.
If you took all the phages in the ocean and lined them up, it would go farther than Alpha Centauri.
Hey, that's how we'll get there.
That's how we get there. We walk on the phages. All right, so they start with a reporter, not a press person, but an enzyme that makes light, luciferase. Right. This is from fireflies, right? It could be from fireflies or what was the other luciferase we used to work with? Fireflies.
Ranella? What's
Ranella?
Frog. Yeah, Ranella's a frog.
No, really?
Yeah. Ranella.
I think you're thinking of Rana pipiens.
Ranella luciferase. Let's see where that's from. I thought it was a frog. I could be wrong.
I think it's some
plant.
What is Ranella?
Some other sea.
A sea pen. Okay, it's not a frog. It's a sea pen. A sea pansy. Okay, sorry, it wasn't a frog. Thank you. But it's not a firefly.
Is that a bryophyte?
I don't know what it is. You can look it up. I've diverged enough. It's a protein which it will make light, and you can measure the light with fancy, expensive instruments. Actually, they're not so expensive anymore. So they have the protein that is called nanoluc because it's a small version of the luciferase that we used to use. And they can take 13 amino acids from nanoluc and stick it on another protein, and that's alpha. That's a alpha part of Alpha Centauri, right?
And Ranilla is a cnidarian. So it's related to jellyfish.
Oh, that helps a lot.
Well, I understood. So cnidarians are jellyfish.
Okay.
Yeah,
I'm good with that.
In fact, when you, it's kind of like GFP, which was from a jellyfish.
Which is also
fluorescent. Yeah.
Right.
That's from a, is that from a cnidarian? I think it's from a ctenophore, which is the other kind of jellyfish, the ones that don't sting. But
so you take 13 amino acids of nanoluke, you can fuse that to one protein, and then you can take the Centauri part and put it somewhere else. When the two of them come together, you will get light. So this is very cool. This is called alpha complementation, right?
Right. So this is like beta lactamase.
yes.
And okay, but so the take-home message is you can divide this protein up into two pieces, and they can come back together in trans and function.
Right. Yes.
Yep.
So it's the 13 amino acids and then 19 kilodaltons, which is somewhere on the order of 172 amino acids. So a really big chunk, and then this little alpha chunk.
Right.
And
So
you can have
apart,
them. Yeah.
When they're apart, there's no light.
Correct.
When they're brought together, there's light. So, for example, you could take IRF3, which is normally cytoplasmic. You put the alpha fragment on it. When it goes in the nucleus, well, then you've engineered the cell to have the large fragment in the nucleus. They put a nuclear localization signal on it, so the protein goes in the nucleus. And then when IRF3 goes in, they will find each other. Dixon, how do they find each other? Well, he's muted,
Delightful.
but he said delightful.
They find
Delightful.
Those are the worms that find each other. Schistosomes, right?
Yes, and elephants in the tall grass.
Yes.
Two schistosomes infect a person, a male and a female, and they find each other so they can mate and make eggs.
In the
And I
liver.
asked Dixon, how do they find each other? And he said, delightful.
Well, that's right.
So these two proteins will interact, and then they will make light. So that's the assay. And you can do this for every
So in this
IRF,
case, they
right?
literally find each other delightful.
Yeah, that's right.
Oh.
Delightful.
Well, they're making light of the situation.
That's right.
Basically.
So I'm thinking
another
about this.
title.
But
There
Alpha
is-
Centauri, I don't think is a binary star, so the naming is a little off here.
So this is random finding each other, right?
Yes.
The IRF goes in and it's just moving around the
Well, they
nucleus.
have the two pieces. I think would have an affinity for each other, and
But
so as
you're
they
just
move
talking
around
about
through,
Brownian motion.
yeah, Brownian
Yeah.
motion and
Yeah,
how do
yeah,
proteins
exactly right.
bind? It's
Yeah,
they're-
part of, as I understood it, part of the development of the assay is making it easier for them to find each other. Okay,
okay.
That's
because
correct.
that's when they
That's
stick
correct.
the alpha bit
Yes.
on another DNA binding protein, because that's where the IRF wants to go.
Right.
Right?
So they
Correct.
put it in the right neighborhood.
Yes,
And then
exactly.
they're gonna stick
that's
together.
right. That's exactly right. So they test this out. They use IRF3 with the alpha fragment, and it just takes cells that are, so you have to engineer cells to make the alpha and the Centauri fragments, right, in the cytoplasm in the nucleus. IRF3 is normally cytoplasmic. And then if you add something that's sensed by RIG-I, which happens to be Sendai virus, and then the IRF3 goes to the nucleus. So Sendai virus you can buy if you don't want to make it, if you don't want to grow it up. It's a paramyxovirus, and it's a really good inducer of interferon. And so when they add Sendai virus to these cells, the IRF3 goes in the nucleus where they can see it. They do bioluminescent imaging, and you can see that the signal is now in the nucleus, whereas before Sendai, it was in the cytosol. So it works. I can see them, planning this for months, and then all of a sudden it worked. That's very
Yeah,
That would be
satisfying.
a great
it
day.
worked,
Yes.
right? Because you don't know if it's going to work.
Right.
And if you're trying to convince your supervisor, you know what they would ask you? How do you know if it's going to work?
Right.
You don't know until you try
You
it.
do the experiment.
That's why we call it research, right? Or an experiment. Anyway, so now can we use this
Actually,
to
we
do
call
drug-
it research because we do it over
and over. That's right.
You
again.
search and you research and you research again.
The experiment is because you don't know until you do the experiment. So can this be part of an assay? First of all, they get rid of Sendai virus because it's not great. they say every virus stock is variable, so let's do something else. So instead of adding sendai, which is basically your positive control to activate the interferon system, they use what's called a 2-card construct. So this is part of RIG-I that will stimulate phosphorylation of the IRFs. So if you put 2 of them together, this is a constitutively active. It constitutively activates phosphorylation of the IRFs, and so they see, okay, this works really well. We can control that, and then, as Rich said, they say, there's just a very discrete puncta of luminescence, and doesn't look like much of the nuclear IRF is near the Centauri, right? Because they have to diffuse around. So they fused SEN in the nucleus to this transcriptional coactivator, Crab binding protein, which is needed, right, for the promoter activation. And so then you get a nice increase in luciferase activity. So now they have this.
So this is all just building the assay to find the thing
assay,
that they want to look for.
And I like this part. I thought it was very cool.
Yeah.
Okay, so then does it work? Proof of concept. They did some experiments to say when is the best time to add the drug, and we don't have to go into that. And then they have an inhibitor of a kinase that phosphorylates these IRFs. It's called MRT67307.
Ask your doctor about no.
It just rolls off your tongue. And in fact, so basically, they add this to cells in their assay, and this inhibits the luciferase signal because it is inhibiting phosphorylation of IRF3, so it can't get in the nucleus. You need to be phosphorylated to get into the nucleus. So it shows you that the luciferase you see is dependent on phosphorylation of IRF. And then they wanted to know, would any kinase inhibitor do the same thing? In other words, is there any specificity to this system? So they have a panel of 21 additional kinase inhibitors that they put into their assay. Again, just adding this 2-card to turn on the interferon system. And they find that of all those 21, 2 kinase inhibitors, which happen to inhibit the same kinases that phosphorylate IR3, also block their luciferase. So it's specific. And now they look at, in this screen, they had other inhibitors. Staurosporine is a non-selective kinase inhibitor, so that decreased the signal. It's non-selective. Okay. Rapamycin, which is a inhibitor of a rather important kinase. And then gilteritinib, which I'd not heard of, but it turns out it is a inhibitor used to treat people with, what is it, AML, acute myelocytic leukemia. So that drug inhibited the IRF3 signal. It was unexpected to them. Also, remdesivir inhibited the signal. Isn't that funny? This is a nucleoside analog.
What about hydroxychloroquine?
They did test it, actually.
They tested it. And
I'm sure they did.
Yeah, they did, and it didn't work.
Yeah,
it didn't work. But ivermectin, yeah, that's in here. We'll get to it.
Yeah.
Of course.
I didn't actually know this had SARS-CoV-2 in it, but it turns out that there is substantial SARS-CoV-2. All right, so they think the assay is now reproducible, specific, get a good signal. You could do this in multi-well formats, so that's good for automation. So now let's do some experiments. They have a bunch of viruses that they're going to test here. That includes SARS-CoV-2 influenza virus and West Nile virus, all different virus families. So they take and they infect their cells, their reporter cells with SARS-CoV-2 at a low multiplicity of infection, and IRF3 didn't go in the nucleus, interestingly. And they say probably it's antagonizing in some way IRF3, which others have published. So you infect with SARS-CoV-2, and you would think IRF3 goes in the nucleus because infection is being sensed, but it's not. And so then they have an inhibitor of one of the SARS-CoV-2 proteases, which is called GRL0617.
Did they say that somewhere? Or did you just have to know that, what the GRL0617 was? Because that threw me for a loop.
It says it here. It says it's an inhibitor of
I had to search around to find it, yes.
the papain-like protease. So interestingly, that partially restores nuclear translocation of IRF3. So in other words, you infect cells with the SARS-CoV-2. IRF3 doesn't go in the nucleus according to their assay. If you add this drug, now some of it goes in the nucleus. Not all of it, but some of it.
It's like the virus is squelching the innate immune response.
Exactly right.
But you want to see the effect of the innate immune response, so you stop the virus from doing that, and then you can see the effect of.
Correct.
This is we're not talking therapy here, okay? We're talking trying to figure out what's going on.
Yeah.
Mechanism, yeah.
Now, here's the ivermectin result, which I don't understand. They treat cells with ivermectin, which, among other things, is an inhibitor of a karyoferrin. Karyoferrins are proteins in the nucleus that are involved in import. So if IRF3 is getting in the nucleus, which it does when it's phosphorylated, it's got to deal with karyoferrin. It's got to interact with that in some way. So ivermectin would be inhibiting IRF3 from coming in the nucleus. Yet they find that pretreatment of cells with ivermectin improves translocation of IRF3. So I don't get that. If it's an inhibitor of karyoferrin, how is it improving translocation? And they don't explain it. So I don't know what's going on there. But ivermectin doesn't work for COVID, so please don't take it.
No, don't.
Pretreatment of cells with interferon alpha gives them more signal because it increases the sensors, and it's got an antiviral effect on SARS-CoV-2, so it gives you more IRF3 in the nucleus. All this is to try and tweak their assays. All right, then they decided to look at the other IRFs. So far we've looked at IRF3. So they make Alpha Centauri for IRF 1, 5, and 7. So now you got 1, 3, 5, and 7. So you got 4 different cell lines. Now you could infect them with all the different viruses. And what they found was surprising in that not all viruses activate the same IRFs. So Sendai only did nuclear translation of 1, 3, and 7, not 5. Influenza virus only activated 3 and 7. And SARS alone was poor at activating any of the IRFs, unless you add this drug that inhibits one of the viral proteases. So apparently SARS-CoV-2 gene products are really messing with the innate immune response.
I want to touch on a little technical subtlety here, which is, if I understand it correctly, all of this expression stuff is being done by transfection.
That's right.
Okay, so that when you do an experiment, you're transfecting in the CBP sen, you're transfecting in the IRF whatever alpha, and when you're doing C-card, you're transfecting that in too. And they talk later on about how they threw out anything where they weren't getting transfection efficiencies greater than 30%, which is, sort of standard. I think getting good transfections is not necessarily easy. Seems to me that in the long run, you would want cell lines that would do this. Certainly, if you're screening a bunch of cells and screening a bunch of IRFs, transfection makes sense. And I don't know if there's, of course, it could be that if you tried to make a cell line, it wouldn't be stable. Could be that these things would be toxic in the long run or something like that, so that you couldn't do it that way. But this was running through my mind as I was looking at the
I
whole
agree.
thing.
I think you'd want a stable for the actual assay. Yeah.
Well,
Unless
you'd
there's
want
a problem.
you'd want a stable line for the initial screen, and I think you'd want to go to primary cells for follow-up screens,
they
which
talk
is something
about
I think
that,
they mentioned in the discussion too,
in the
because
discussion,
using all these immortalized cell lines, I mean, HeLa cells we've talked about; these are far removed from anything you're going to encounter in the body. So you'd want an initial screen that's a stable line that you don't have to do all these transfections in, and then follow up with something that's a little more realistic.
All right. So as I said, they have cell lines, not cell lines. They do transfections, as Rich said, with the 4 IRFs with the Alpha Sen system, and they look at West Nile, influenza virus, Sendai, and SARS-CoV-2, and they get different in their assay. And so, for example, Sendai pushes all the IRFs into the nucleus. Influenza just does some, and SARS-CoV-2 doesn't push any of them in the nucleus. So it's kind of interesting that there's a differential effect. They say different IRFs respond to different viruses, which could be good therapeutically if you can specifically hit one of those, right? So you don't have to have a broad treatment.
Well, one of the things I thought when this came around, that the IRFs respond to different viruses in different ways, is, why do we have all these IRFs? Well, this is why.
Yes, that's right.
Because we got a lot of different viruses.
Right.
We kept evolving them because the viruses kept stopping them.
Exactly.
Yes.
Yeah, actually, that's probably right. The viruses kept putting up countermeasures. So, okay, we got to have a new IRF.
So then they say, let's go back to those inhibitors that we previously used and see how they work in our assay. And they're using SARS-CoV-2 now. A whole panel of inhibitors like gilteritinib and hydroxychloroquine and remdesivir and a whole lot of others with not names that you would remember. And they say the most striking hit was gilteritinib, which increased immune signaling, so the IRF going into the nucleus by over 300%. And then they have some other molecules that also work. This is an interesting one, scutellarine, a naturally occurring flavonoid isolated from traditional Chinese medicine that was shown to inhibit SARS-CoV-2 and CoV and CoV-2 enzymes in vitro. So it actually has, cool. It allows influx of IRFs into the nucleus. So they focused for the last experiments on gilteritinib. They want to know how it works. And they find that it's actually, so besides this assay where they're looking at luminescence, they also say, does it inhibit virus reproduction? They do a plaque assay.
Yeah.
And it's quite a strong, they have 50%, IC50, the 50% inhibitory dose is 0.13 micromolar. So it's a good inhibitor. Remember, so it allows, gilteritinib allows IRFs into the nucleus in their assay, and it's also antiviral. So they want to know how this is working. And IRF7 is the IRF that gave them the strongest effect of gilteritinib. So they think somehow that's the target of this drug. So they show in a series of experiments that gilteritinib stimulates the expression of interferons, and if you use an antibody against interferon receptor, you reverse the antiviral effect of gilteritinib, and then they find that it inhibits other viruses as well. So it inhibits West Nile and HIV and influenza virus. So this is, again, gilteritinib, this kinase inhibitor. And so, as I said, gilteritinib is used in cancer treatment. It mainly inhibits two protein kinases. They're called receptor tyrosine kinases, called FLT3 and AXL. So they knock down the genes, the mRNAs for both of those tyrosine kinases, and they show that when you knock down AXL, you reduce the effect of gilteritinib, suggesting. So remember, the effect is that when you add gilteritinib to SARS-CoV-2 infected cells, you get more IRF7 going in the nucleus. That movement, which they call immune activation, depends on AXL, which is a kinase that is one of the targets of gilteritinib. Gilteritinib. So, just so you know, this drug is used to treat AML. The most frequently mutated gene in people with AML are in FLT3 and AXL. FLT3 is a kinase, and AXL is needed for activation of that kinase and has a role in the disease. And so that's why this drug works for AML. And so AXL also appears to be important for the immune activation of the inhibitor gilteritinib in SARS-CoV-2 infected cells. Okay, so gilteritinib is not their end product. They just show that it works in their assay and they can figure out how it works. But now one assumes that they will do a massive screen of small molecules.
A whole bunch of small molecules, yeah.
Looking for those that will allow IRF7 or maybe the other IRFs to get in the nucleus in SARS-CoV-2 infected cells, right? So specifically activating those so that you can get a good interferon response in infected cells, not everywhere, but just in infected cells.
Right. And this is a good point to just reiterate. This is a proof of concept and mechanistic study. Do not go browbeat your doctor for gilteritinib to treat your COVID-19. And you can look up gilteritinib side effects if you want a little primer in why you might not want to take that approach.
But would be interesting to know if people who are getting gilteritinib, because they probably get an infusion consistently, right, Dixon?
I think it's dosed orally.
Orally?
I don't know about the treatment for leukemia. That's a totally different animal.
But they must be getting multiple.
They're getting it's ongoing dosage.
Yeah, I wonder if they have less SARS-CoV-2 infection, right?
That's a good point.
You might be able to mine those data.
Although somebody with leukemia is going to have some immune system issues potentially that might override anything you're getting from the drug.
I'll say.
Anyway, I thought it was a cool assay that they developed, and it seems to me that you could use it to look for drugs that do immune activation, is what they call it. Allow these IRFs to get in the nucleus of infected cells and then turn on interferon synthesis.
And getting back to the original introduction of this whole thing, the idea is if you had these drugs, you could potentially try them against novel viruses that emerge if it looks like a failure of the immune response or misguidance of the immune response is part of the syndrome for that virus. Maybe you could put it back on track by selectively inducing interferon in infected cells with some drug that does that.
They also
this is the
suggest
pathway to discover that.
If a new virus emerges, you could do this assay very quickly and maybe get some candidates.
Yeah.
So I'm riffing on Scutellaria here. So it comes from a plant called, what is it? Scutellarian. This is the one that's from Chinese traditional medicine.
Right.
So the plant that it comes from is called Scutellaria lateriflora. It's a flavonoid. And I'm thinking about, traditional medicine. I'm thinking, I'm going to come in sick and somebody's going to say, here, take this plant extract and it's going to stimulate your immunity and help you out. And I'm going, yeah, right. Well, maybe so. Isn't that interesting?
You need to do the experiments to show it, right? And they did here. It does. Exactly. It works.
So doesn't vinblastine and vincristin come from the periwinkle?
Yes.
So I would take the drug, Rich.
Yeah, I'm all over it. This is great.
So, Scutellaria is a genus of flowering plants in the mint family.
Yeah.
Also known as skullcaps. In North America, Scutellaria latifloria became a common treatment in America for rabies.
Wow.
Oh, really?
Today, it is still a popular medicinal herb.
Rabies?
I'm going to bet that it doesn't work very well against rabies.
No.
Just guessing there.
I bet it's so it depends how much of this compound is in the plant, right? If it's a small amount, it's not going to have any effect.
Right.
And you'd have to purify it and make it, and then it would work because this drug is obviously purified.
And Wikipedia tells us that that's a dead link.
Okay.
So we can't find that.
We can't
follow that
trace
up.
All right. Dixon, can you take that first email?
Yes, I'll be doing my best, although I'm having some voice problems
It's
here.
a short one, you know?
Yeah, I got it. Jessica writes, as a young nurse years ago at a Midwestern University hospital, I watched numerous interns and residents examine wounds, infected and otherwise. Not only were they really universally ignorant or dismissive of sterile technique, but almost every male one accidentally dragged his necktie into the wound. A bow tie is much more safe for patients' care, and ID docs like Daniel Griffin are hip to this. That's a good observation, actually.
Yeah, dude,
I'm
Dan
wondering,
only wears...
pediatricians are big bow tie guys, and that's probably because the kids are grabbing your necktie all the time.
And choke you. Yeah.
And pathologists traditionally wear bow ties because otherwise you get your tie in whatever pathology sample you're working with.
So, and this came up because of the TWiV that Vincent did with the science artists and talked to Michelle Banks about silk and maybe making silk bow ties.
Yeah, that's right.
This reminds me of one of these. It was one of these checklist in medicine studies that actually focused on basically wound infection. Or actually, no, it was, I think, infections from sticking in lines, okay, where they had a checklisted protocol for doing this that mostly focused on the appropriate sterile technique. And they also gave nurses in this study the authority to call out whoever was doing the procedure, like the docs or whatever, which is, part of the problem.
Yeah.
Okay. She says here, universally ignorant or dismissive of sterile technique, right? And the study showed that if you apply a checklist and have people adhere to it strictly, that the infection around lines goes way, way down.
Alan knows all about checklists.
A century of aviation was not wrong. Wow.
Exactly.
What do you know?
Nice laminated checklist, right?
Yeah.
There you go.
Yeah, I mean, the problem in medicine is that you have so many different procedures that you would have to come up with a checklist for. But that's also an argument for them, because one of the purposes of a checklist is to do stuff that you don't do frequently enough to have memorized. Yeah. And you don't want to forget, what was the 12th item we were supposed to do here. So,
Kathy, can you take the next one?
Sure. Luke writes, hello, TWiV team. It is currently hotter than it ought to be in Columbia, Missouri, but that just means that I have to wake up earlier to water the tomatoes. I have a question regarding a recent paper discussed in TWiV 911 regarding EBV nanoparticle vaccines. In particular, one point was brought up in reference to Figure 5, wherein viral DNA was observed in some individuals of the 60-mer group, and it was suggested that this implies sterilizing immunity was not established. However, since the mice challenged here were not producing their own antibodies, 1, is there any reason to suspect this conclusion to be different in a model that could both produce its own antibodies and permit infection? 2, how sterilizing does the vaccine need to be? Yes, here we are trying to avoid establishment of a latent infection, but can the immune system realistically identify a microbe as a problem, initiate an adaptive immune response, and deploy the troops before a single cell is infected? Should we approach this infection from a different angle, mayhap with improving CD8 positive T cell recognition of latently infected cells? 3. Additionally, it seems that the study was cut off short as the virus could simply have been in a latent phase and only able to spread once the antibodies from passive immunization dissipated. Thank you all for your time, and I really appreciate what you folks do. Luke.
Heavy-duty questions, Luke.
Good question.
That's
Yeah.
pretty well thought out for an electrical engineering student. That's for sure.
Chemical engineering. So Luke
I'm
is
sorry, chemical
identified as a
engineering.
PhD candidate in chemical engineering at University of Missouri. Good questions.
So I think it's a good point. You're never going to be able to prevent a single cell from being infected, right? But maybe you could improve killing of infected cells by focusing on T cell epitopes and not rely on antibodies only, right?
Yeah.
Because there's always going to be a delay in memory recall. So I think that's a good point. I think it's funny how sterilizing it has to be. Well, sterile is either sterile or not, right?
Right.
So I don't know. It's a good question.
But sterilizing immunity is a huge bar.
Yes.
It is.
And in particular, as he rightly points out, in a situation where you're dealing with a virus that establishes latency. Because once it establishes latency, then you got a problem.
I just don't know how good how much you have to prevent. Can you get away with some cells infected or not? But that would be an experiment I think you could do. But it seems to me they don't have a great animal model, right? Because they have to engraft these mice with human cells, and it's not great. And they have to passively give them antibodies as well.
And could this conclusion be different in a model that could both produce its own antibodies and prevent infection? Yeah. Okay. It's all about the model. All the models are going to have they can't have a lot of variability.
Maybe the experiment to do would be, instead of passively giving them antibodies, to passively give them T cells.
Sure.
And see, but their readout is PCR detection of DNA in the blood. So it's going to be really sensitive. What you want to know is if you establish latency. So you need a latency model, right?
Yes.
So they're all great questions, but I don't think we have the ability to address them. Alan, can you take the next one?
Sure. Martha writes, Dear Vincent and the gang, is polio rearing its ugly head once again? Coming back from the dead, if you're in that specific belief system, virus equals living? Of course not. The fact that I know this has to be unanimously attributed to the excellent educational work of microbe.tv since its inception in 2008. In fact, episode 2 has it all for the uninitiated, and it will forever remain one of my favorites, as it showcases how fundamental basic knowledge about the virus needs to guide appropriate public health measures. In this light, it is forever frustrating that journalists and the media, and sometimes other experts, do not seek out the information about what they report and just fearmonger on the internet. The death of expertise, indeed. Luckily, New York doesn't test its sewage, right? Because look what happens nowadays when you find a virus. Or I'm having this the wrong way around, and some brilliant mind spread the angst to have reluctant parents go get the bloody vaccine. I think it might be timely to pick up the educational torch on the next episodes to highlight the upsurge in polio all around the globe due to this other little virus that's been basking in the sunshine for so long. I'm sorry this became a bit of a rant, so let me end with another expression of gratitude. I love, love, love TWiV, and I hope it will never end broadcasting. Cheers to the team, Martha. Thank you, Martha.
So TWiV 2 is entitled Polio is Not Dead, September 26th, 2008.
Wow, I'm impressed, Martha, that you listened.
Yes, I remember it well.
Yeah,
and that
Vincent
was
Racaniello and Dixon Despommier.
Wow.
Yeah, just Dixon and Vincent, yeah.
Yep.
I'm just so impressed by your email, Martha, that you understand this, but also you remember what we talked about so long ago.
Well, you could also go back to the archive and listen to it.
That's true. That's very true.
She says it's one of her favorites.
One of her favorites. So yes, that probably has been going on a while.
I don't think there was some brilliant mind who spread the angst to get parents to get vaccinated. I don't think that works. But this idea that SARS-CoV-2 has reduced polio vaccination, we did address slightly earlier, right?
Yeah.
You mentioned that. It is a problem, yeah. It's inhibited vaccinations. The death of and also Martha listens, obviously, because the death of expertise was my pick a couple of TWiVs ago.
Mm-hmm.
I have to say, I am always impressed when people do their work, do their research, right? It's not really research, but just pay attention. How about that?
Yes.
Pay attention.
They participate.
They participate. Martha is clearly participating. I mean, she even does the living or dead thing. She's got it all. This is
She's
Martha.
paying attention.
I'm very impressed, Martha. Excellent.
And she's also a supporter.
Proud
Yes.
supporter
Microbe
of Microbe TV. Thank you so much for your support. We will try and keep broadcasting forever.
Yeah.
At some point, it will not be in our hands any longer.
That's right.
This is all true.
Rich, can you take the next one?
Charles writes, hello, Twivers. Too damned hot in North Carolina today. I hear you, Charles. If you need to be pissed about the intersection of science and politics, read this. And he gives a link that I'll come back to. How can there be a licensed family doc that is a vaccine skeptic? State boards should have suspended their licenses a long time ago. Later, Charles. The link is to an article, a little bit in MedPage today, that's titled Doctors Candidate Threatens Minnesota Medical Board. And it highlights an MD by the name of Scott Jensen, who's Minnesota's Republican gubernatorial candidate, who's threatening to retaliate against state medical board after it opened an investigation into him for the 5th time, according to a report on AP. And this guy is a vaccine skeptic. And he's encouraged civil disobedience over mask mandates and promoted ivermectin as a COVID treatment. And he won the GOP endorsement to run against the incumbent governor, Tim Walz, and has criticized his opponent for his pandemic response. Well, you know, go figure. Here we are. Crazy. Crazy.
I don't know anything about licenses and what kind of activity you would have to do to get your license suspended. You have any idea, Alan?
There are a variety of things. This kind of thing could certainly be brought up with the board as, bad for public health. And usually doctors the board doesn't go after doctors for making statements about things because usually they don't reach this kind of scale, I suspect. Most board actions are things that the doctor has some pattern of behavior or has demonstrated their incapacity to practice medicine in appropriate, ethically acceptable ways. I mean, you get the picture of how these things usually evolve. So I don't know what the exact beef is here, and I'm not planning on digging into it. Except to say, listeners in Minnesota, vote this guy into the dustbin of history and make sure that does not get.
Well, it is a part of the theme here is that the Minnesota State Medical Board has investigated this guy now 5 times.
Right.
Okay. So they aren't happy about it.
No. And I don't know exactly what he's done, but it seems like he's quite a piece of work and probably should not be practicing.
I mean, the studies are in. Ivermectin is not useful, whether you give it early or late. Doesn't help. I don't get it. This reminds me of, the Surgeon General of Florida who said COVID is a zero issue in kids, right?
Yeah.
How can you be a doctor and say that?
You can't.
I don't get it.
No.
Well, you can.
Right. But
well,
I mean, it's just lying for certain purposes, and I just don't know why you would put people's health at risk.
There's a Senate candidate who was formerly associated with Columbia University who has an MD,
Yes.
You know, so who has made repeated statements over the years, even before going into politics, that are just completely at odds with medicine.
So, some of his colleagues have said, "I hope he gets elected. Get him out of medicine.
Get him out of medicine." Yeah.
Oh no. He won't get elected. I can tell you that. He will not.
Gosh, I hope not.
Nope.
So one of the things this makes me think is buyer beware, even in medicine. You know,
Yes.
It took me a long time to catch on to the fact that I was the guy who was responsible for my health.
Yeah.
Okay. And you
That
go
is
to the
true.
doctor and you can find good doctors, and you can also find doctors who, you know, maybe are not quite so good. And you got to be resourceful to find the right. You got to take some responsibility for your own
Although
solutions.
I will say, and I am biased being married to a doctor, but I do think the majority of them are doing the best they can with the tools available
Absolutely.
to them. We do have a badly broken healthcare system in this country, but that's not primarily because of the doctors. That's because of everything around the doctors.
Not only that, but in my experience, the majority of them are extraordinary people with an extraordinary sense of compassion and service. Okay,
because they didn't go into it for the money. These are people who
Right.
went into it to take care of individuals.
Yeah.
Right. I was just going to say that I think, at least with some of the older generations, I think this was the case with my parents, is that they tended to take whatever physicians would say, you know, as the gospel truth, and were really reluctant to challenge them.
Yeah.
And I think it's important to know that, yes, they're doing the best they can, but they are human. And that's why getting a second opinion is important. And then the extreme can be people like this who've been called before the medical board 5 times. I mean, once, you know, might be some kind of spurious or somebody has an axe to grind, but 5 times makes you think that there's something wrong there.
I mean, in medical school, they teach you that vaccines are good, right? Was he sleeping during that lecture?
No, he just chose to reject it for ideological reasons.
Yes,
and that's
correct.
the problem.
Alan's right. Alan's absolutely right.
As Daniel would say, get a new doctor.
Yeah.
Yes.
Yeah.
Yes.
And keep your same governor. Don't put this guy in the office.
All
All right, one more from Moro. Greetings from Buenos Aires, Argentina. The children's book contest prompted me to write this poem. I don't expect to win the prize. I'm not a writer, a poet, or a native English speaker, but I hope this makes the cut. If I'm lucky to get it read on the show, may I ask for Vincent to do the honors? The ending suits you. I had fun writing it anyway. What you do is outstanding. When I wake up, I pick up the phone not to check for messages, but for new TWiV episodes. Outstanding.
It's great.
So here we go. This is a poem called Will Somebody Please Think of the Viruses? Though now we are alive, the final day approaches. They don't need us to survive, bacteria, rats, or roaches. But who among the species will suffer our demise? Who will carry their pieces as they mutate, infect, and splice? For they are part of us and we are part of them. If we're sharing our genomes, aren't we the same? The less of us there is, the fewer of them there are, the viruses for whom there is no reservoir. And so one day we'll find in this humble earth we roam, there'll be no humankind and no human virome. I don't worry about walruses or any animal you pick. I wonder about viruses, the kind that make you sick.
Beautiful.
Outstanding.
Beautifully
Well
done, Morrow.
done, Vincent.
Oh, thank you. Thank
And
you.
well read, yes.
This is great, Morrow. That's just beautiful. I love it.
That should win a prize.
Yes, we
That
will
should
send
win a
you.
prize.
Morrow, send your address, twiv@microbe.tv, and we'll ship you out some of the books by Pamela Jane. And any others want to try your hand, we have quite a few books. Love to have you. I just love it. He
great.
said, I'm not a writer, I'm not a poet, but you did a great job.
Yeah.
Yeah.
No, it's very...
And now that brings us to Picks of the Week. And Dixon, what do you have for us?
So, continuing on with my desire to entertain rather than to inform about virology, which I could certainly do, but it would be all wrong, obviously. We had a discussion, Vincent and I, about my likes and dislikes. And one of my extreme likes and passions is music, and in particular jazz. I was a young person when I was first exposed to jazz. And as I grew up and eventually entered high school, we began to go over to places like Birdland in New York City. We couldn't buy any drinks, but we could sit and listen to the music. And so this band, the Count Basie Orchestra, and I'll give you my picks of the top 5 bands, the top 5 small groups, the top 5 instrumentalists. You tell me when to stop. Count Basie used to play Birdland at around Christmas time, and a lot of us had off then. So we'd go over in a group, like 10 or 12 of us, and go into the portion of the place called the Bullpen and sit and drink ginger ale and listen to this fabulous music. It was just fabulous. And so my pick for the top jazz band, big band, is Count Basie between the years 1952 and 1965. The signature song that they became known for was called April in Paris. And you can see it listed at the bottom under the link. And listen to it.
Excellent.
And the signature album, of course, was called April in Paris. So if this is okay, I will continue. If not, we can
Go
pick
for it,
another
Dixon.
subject.
It's great.
This is great.
Yeah.
Oh, I
All
love
right,
it.
All right,
I
we'll
love
do
it.
that. And you get the song also. You do get the song. So you can judge for yourself whether you like it or not.
So you're going to do your 5 big bands and 5 other,
Then
right?
5 small groups, and then 5 trumpet players, and then 5.
Great.
Cool. Very good.
Yeah, I'll do that if you
Now-
like.
Dixon,
This is totally appropriate picks for the New Orleans originated TWiV co-host. I like this.
well, that's your interest in jazz came from growing up in New Orleans, right?
I didn't grow up in New Orleans. I was born in New Orleans. I grew up in San Francisco
When did you leave New Orleans?
when I was 1 year old.
Yeah, so it was nothing, right? No influence
It
whatsoever.
was in the
No,
water.
genes-
it was
the
in the
genes were
air.
there.
It was in the water. He was
was
exposed to it
especially in the food.
prenatally. Yeah.
That's right. That's right.
And you do play the trombone, right?
I used to. I haven't played it recently, but yes, I did.
I saw you
Yeah, I
play
had
it at Coogan's years ago. You remember?
I had a wonderful life as a high school and then as a college student, playing in various jazz bands, and met some interesting people. That's all I can say.
Great.
Cool. No, it should be fun. I'm looking forward to it. Kathy, what do you have for us?
I picked something that was first, I found it as a lay article in one of the University of Michigan news things. It's about a collaboration of Aerospace Engineering and the University of British Columbia, some zoologists. And they're looking at bird aerodynamics and inertia. And so it's really cool. There's a Nature open access paper that they all wrote together. And then there's a video that kind of tells it all. And for the second time I watched it, it had the closed captioning appearing, and I thought that was helpful. But basically talking about how you can, study how birds fly and how they always thought that the evolution of birds was toward more unstable gliding, like from dinosaurs, which were, of course, early forms of what are now birds, to birds now having more unstable gliding. But actually that the key to this all is the switching between stable and unstable gliding. And so it's a little bit heavy on the aerodynamics and the physics kinds of things. But the gist of it is that it could give some help to what they call, what's their acronym?
UAVs, unmanned aerial
UAVs,
vehicles,
which,
sorry.
I think of as
Drones.
just like drones.
Yeah.
And the video shows something in what I consider to be the drone cage at Michigan up on the engineering campus. But so it's just a really interesting mix of engineering and biology, I thought.
Cool.
So as I just skimming this as you're talking, it seems to me a lot of this is about how these birds can tweak their wing shape to go from one mode of flying to the other.
Yeah.
And what that reminds me of is, if I'm not mistaken, the original Wright Flyer actually tweaked wind shape to maneuver the airplane. It turned out to be not the best way to do it for that, but it makes me, because I know the Wright brothers spent a lot of time studying birds.
Yep.
And I wonder if they were adapting that sort of thing that they learned from birds to the Flyer to try and do the same thing.
Yeah,
Leonardo da Vinci preceded him by 300 years.
Right, so
and he also studied birds.
The Wright brothers
picked it up.
The wing warp concept on what birds do as they turn.
Yeah. Okay.
And it turns out that there's a limit to the flexibility of materials that can make an airplane that won't fly apart. And so you're better off having separate panels like flaps and ailerons to do this sort of thing. But this is really cool that they've, I mean, they're basically mapping the flight controls and the aerodynamics at a very detailed level. And I'm not going to wade into the physics of this, but this looks like a really cool study.
I love the title. Birds can transition between stable and unstable states via wing morphing. And I just want to put viruses can transition between stable and unstable states via conformational changes, you know? It's just same thing.
Capsid morphing.
Yeah.
Capsid morphing. It's so cool. I mean, if you think about it, what they say is they compared 22 species across the full range of elbow and wrist flexion and extension. So that's how they have different shapes to their wings. It's so cool.
Yeah, because wings are hands.
Mm-hmm. Yeah. So, they show where the elbow is and where the wrist is.
And there's a lot of math.
Yeah.
A lot of equations for this. A lot of bird-specific math.
Yeah, the first paragraph has got 2 equations.
Yep.
I picked this based on the video.
Okay, sorry.
And the article. And then I thought, oh, look, the paper itself is open access. I'll get that too. And
I went right to the paper to see that, but I should probably look at the video. Yeah.
Yeah.
Very
cool. Neat. Rich, what do you have for us?
So several weeks ago, I sort of refreshed, using a pick, refreshed the discussion of Artemis, which is the mission or series of missions that are intended to return humans to the moon. And I had not done my research very well on that. And the question came up during that discussion of, what are the details of how is there going to be a lunar orbiting piece of the spacecraft and a lander like before and that kind of stuff. And I didn't really have a good answer to that. So I got an email from a listener named Ellis, who's actually here in Texas, down the road from me near New Braunfels, who really got off on this because I guess he's a space program freak, and pointed me, actually, his initial point to me was that the NASA is putting out bids for people to design or submit designs for different landers. Okay. But that got me into what the whole strategy is going to be. And central to that strategy is this thing called Gateway, which is going to be a permanently lunar orbiting space station.
Yeah.
Okay. That will have many functions. One to live aboard and study the moon. Another is basically a platform out of Earth's influence at all for missions beyond, and also as the way station to the moon. And so my pick is a link to NASA's description of Gateway that shows their concept of what that's going to be built of and how it looks.
That's a good pick.
That's just very cool. Awesome.
So the Gateway is kind of like the command module of Apollo.
Yes, it's a command module on steroids.
Yeah. And then they have human landing systems, which is very cool.
They're under development. I think they've probably got a couple of designs already. But the point of Ellis's thing was that they're asking about more. Also, there are rockets that may take off from may take off from Padre Island down here in Texas, and Ellis wants us to go watch some of these things launch, which strikes me as a great idea.
Yeah.
They're about to send an unmanned mission all the way
Yes.
around the moon now,
Right.
just to see that things work out okay.
Yep.
So if you go ahead.
No, I was just going to say, so Rich, I don't see a Canada arm on the gateway.
That's right. There's a European Space Agency and JAXA, which is Japanese Space Agency, I think. SpaceX.
And NASA, of course.
Here. NASA.
Northrop Grumman, Maxar.
CSA, ASC, what's that? Is that Canadian Space Agency, I'll bet.
I think so.
Maybe.
Yeah, maybe. So maybe they'll get an arm on it.
Robotic system. And look, there's a little arm.
Yep.
Okay.
Yep.
Okay, good.
I'll be darned.
We're going to be armed by the Canadians.
So to get there from Earth, the rocket goes up, and then it's got to dock with this gateway system.
Yeah,
right?
I would assume so. Yep.
Neat. Good stuff.
Good stuff.
Very good. Alan, what do you have for us?
I have an article from a site that I think might have been somebody's pick on TWiV. So the site is Reasons to Be Cheerful. Did somebody pick this at some point?
No, I haven't seen it.
Okay, then consider this a double pick. Reasons to Be Cheerful is a news site started by David Byrne a few years ago, who is best known as having founded the band Talking Heads.
Talking Heads, right.
So you may find yourself on a website and this one article.
And you may find yourself in a house. And
yes,
you may find yourself.
Yes. How did I get here? And Alan sent you is the answer. Anyway, this particular article got my attention, especially with today's topic. And this just came out today. And I said, oh, yeah, this will be my pick. And it's about overcoming vaccine hesitancy in Nigeria for the polio vaccine, which this has been a fraught subject in Nigeria. And there's a history here that's not just about, people having some knee-jerk anti-vax attitude. There was actually a clinical trial in the 90s. It was the Pfizer meningitis vaccine in 1996, which did not go well, and 11 kids died and some got disabled. And this was not a good thing at all, and it created a wave of anti-vaccine sentiment in the country, which was then fanned by political and ideological forces, and a number of members of the clergy started spouting conspiracy theories about how polio vaccine was going to sterilize people, so this created a huge problem, and it's one of the reasons that Nigeria remained for a long time one of the few pockets of endemic polio in the world. But they've been making progress in recent years, and Nigeria is the most populous African country, so this is a big deal. And this article is all about how the effort to overcome that hesitancy has to work with people who are trusted in the community and has to be done on a local scale. And so it's this particular example of they're going to this village, which has had for centuries this king of the village who is recognized as the authority on community matters in the village. And they convinced him of the importance of vaccination, and he got his son vaccinated and then sent out the town crier, which is apparently still a thing in this village, to announce that, the vaccine is available and the king recommends you get it. And the mothers brought their children out to be vaccinated against polio as a result. So this can be done. You just have to manage it in line with what the local concerns are and work with trusted messengers in the locality. It's a very cool approach. One that should be used more often.
Yeah. It's great. I wish it could be.
Yeah.
Well, speaking of talking heads,
which we are.
We are talking heads. The bassist for Talking Heads is Tina Weymouth.
Yes.
When I was a postdoc at MIT, there was a postdoc with Phil Sharp. Her name was Lisa Weymouth. And she was Tina's sister. One day Tina came and everybody
How about
freaked
that?
out.
Oh, cool.
Oh my God, a rock star is to visit her. Yes. And I just looked up Lisa. She's still doing some virology, it seems.
Cool.
And Tina's still playing music.
I didn't realize that Nigeria was the most populous country in Africa.
Mm-hmm.
Yeah.
I'm looking through pictures here. Very cool.
That's right.
It's also the richest.
Yes.
Although that
Yes.
wealth is not widely distributed.
No.
No, that's why they have civil wars every now and then.
They also have good science, relatively good science. Remember, Mavis was from Nigeria originally.
That's right.
Although she wasn't trained there
Mavis.
as a scientist, I guess. Trained elsewhere. My pick is so this is summer. I'm reading. I haven't read in a long time. I thought I would read some books, mostly because I'm traveling more and I read when I travel. And my last pick was The Death of Expertise, and now I'm reading The Tyranny of Merit.
Excellent.
I think you picked this once,
I picked
didn't
this
you?
quite a while ago,
Yeah.
but it's worth repicking.
So it is all about this idea of meritocracy, right? You get what you do. And the book goes through how this arose in society, his religious roots actually, and moves towards contemporary times, and how, most recently President Obama, always espoused this idea in the U.S., you can be what you want to be as long as you work, right? That's what he kept saying, as long as you work hard. And so this gives rise to the tyranny of merit, that the people on top think they earned it,
Yep.
and the people on the bottom feel they failed.
But as Sandel explains in the book, Obama did not start this by any means.
No,
he
he
extended
didn't.
something
No.
that has been heavily promoted since the Reagan administration.
That's
And
correct.
That
And
in
Clinton
those
did it
40
as well.
years, and he talks about the trends that has produced, which is this huge wealth disparity and these ideological divides and all that as
Right.
well.
And then, of course, the creation of the global economy removed many jobs from the U.S. And so these individuals who are, they're not born into the right part of society, and they can't do anything. They can't get jobs, and they feel like they failed, and they're pissed. And the result of that was the election of a person who should not have been president some time ago.
That is one result, but
one result.
globally, this has also been a problem.
But you think, you can do whatever you want, but you can't. It doesn't work. So these are some
unless you are born with extraordinary natural gifts, which I think is the case for
Some
Obama,
people.
who was born into a relatively poor, disadvantaged environment. But I mean, most people are not him. That's
those are rare cases. It does happen, but it's rare. And the fact is, most people, that doesn't happen, and you have inequality.
Yeah.
So here are some good quotes. The meritocratic ideal is not a remedy for inequality. It's a justification of inequality. In an unequal society, those who land on top want to believe their success is morally justified. In a meritocratic society, this means the winners must believe they have earned their success through their own talent and hard work. And that may not often be the case. Anyway, it's very good.
Yeah.
And sad.
I think it's a very important book that people should read.
Yes.
The entire academic community of the world is based on this.
Yes.
It's this kind of the whole thing kind of.
And
turns my
Sandel
stomach a little
talks
bit.
extensively about the college admissions process and how, especially
Getting tenure.
Just the admissions process, because college has become
yes.
this gate that is, if you don't get your college degree, you're going to be probably permanently shafted socioeconomically. And getting into college has become much, much more competitive, to the point that, getting into an elite school is now something that people will break laws to try and do. And then he discusses that problem extensively.
It's also part of the death of expertise.
Right.
The death of expertise has several sources. one of which is the internet, but one of which is college education, which he says is now a business and you can't flunk your customers. So you don't teach students how to think critically and actually do research and so forth. And
what's his alternative?
Sandel has some interesting proposals.
As does the author of the
right. One of Sandel's that I thought was really cool was college admissions, since it's become very much like the grant approval process, where you're making fine distinctions that people can't actually make. He proposes essentially what some other people have proposed for the grant system, which is you'd have a college application process that would triage to the top third, because there's good evidence that you can actually make a meaningful distinction at that point. And within that top third, okay, Harvard's only going to admit 1%. Do that by lottery. So distribute your 1% as a lottery among your top third, which would be fairer, because you're not making these arbitrary decisions, and also would perhaps help bring across the idea that there is an element of luck in the success that you've achieved. You didn't necessarily get here all on your own.
Sounds like lawsuit 101 to me.
Not happening for the same reason that NIH will not do a lottery, because the best funded labs will object because they will lose a lot of
people at the top are the ones in charge, and they're the ones who have the most to lose by the fixing the system. Yes.
Anyway, you should check it out. Tyranny of Merit, Michael Sandel. That's it for TWiV 913. Show notes at microbe.tv/TWiV. Questions, comments, poems, pics, TWiV at microbe.tv. If you enjoy what we do, consider supporting us. Microbe.tv/contribute. Kathy Spindler's at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun. And I learned a lot. And I don't say that tagline very much anymore because I guess
you're not learning.
No, because I thought it was too long compared to everyone else's. But
I thought you were just being selective and maybe some episodes you learn less than others, but I think it's
that's good. Probably true. And there's some episodes I retain a lot less, but that's why I listen to them again.
Dixon Despommiers at Racaniello.org, thelivingriver.org. Thank you, Dixon.
You're very, very welcome. It's great to see two new old smiling faces. Sorry for the nod off.
No problem.
And, it won't happen again.
Well, no, you shouldn't say that because it probably will, but it's okay.
Well, it shouldn't.
We have video editing capability.
Or you have video evidence.
Yeah.
Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove's at alandove.com. Alan Dove on Twitter. Thank you, Alan.
Thank you. It's always a pleasure. And I learned a lot as well. But I just want to say, Vincent, I'm sorry if I stepped on your pick. I know I jumped in there to talk about it.
No, thank you. I appreciate it. I knew that you had picked it, and I was hoping you would...
Great minds think alike.
That's fine. Absolutely. No problem. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.